Okay, so we did the introduction to biological molecules. Now let's zero in on each individual biological molecule. So let's start with carbohydrates. So carbohydrates are sugars, and obviously these are very important to us because we use them for energy. We use them for energy, and we use them for storage, for storing energy, and we use them for storing information as well. Our DNA is deoxyribose. The ribose part of DNA of nucleic acids is a sugar, or actually DNA has a sugar component to it. So where should we start? Well, I guess the most basic use. So you can categorize sugars by the chain of monomers that sugars come in. So a monomer is the basic block of a polymer, of a larger chain. So the basic block of carbohydrates in humans and living organisms is going to be glucose, okay? And that's a 6-carbon sugar. And the basic formula for carbohydrates, the empirical formula, if you will, is CH2O. That's the most basic formula, the most basic carbohydrate. Carbon and water. And carbohydrates basically means hydrates of water, of carbon. Sorry, hydrates of carbon. So you've got carbon with the water. Pretty simple. One thing that you notice as you study these molecules and stuff is that these organic compounds and these organic molecules are things that we find in nature. So water and carbon are just elements that come from stars, basically, are made in stars that are combine themselves to make us. We're just the same as the rocks and the ground and the things, or coal and the things that are around us. Anyways, so carbohydrates, carbon, and water. That's the most basic formula. So a 6-carbon monomer its formula is going to be C6H12O6, and that will follow it's a multiple of the empirical formula. So what does here we go. Here's glucose. Let's look at a glucose here. Can we zoom in? Can you guys see? There we go. There's a 6-carbon glucose. I don't know how well you guys can see that. 6-carbon glucose. And then what you do is you just stitch those together using dehydration synthesis. Remember dehydration synthesis. And if you stitch 2 monomers together, 2 glucose together, you will get a disaccharide. Di means 2. So your monosaccharide is 1, disaccharide is 2. Stitch a few more together, somewhere about, where are we here? Like about 8 or something. Less than, when do we say? When the more than many monosaccharides are joined. Do what I say? Well, a few. So a few monosaccharides chained together is going to be an oligosaccharide. And then a long chain, many of these monomers stitched together, is going to be a polysaccharide. And they all have different functions. They all have functions within our bodies. So like sucrose, for example, is table sugar. And it is a disaccharide of glucose and fructose. Okay? And fructose is a common sugar in fruit. So you stitch those two together, you have sucrose. So sucrose is made of glucose and fructose. And then when we eat them, we use hydrolysis to break that covalent bond that's holding those 2 sugars together. And then we can metabolize glucose for energy in a process called cellular respiration. We take it into our cells, into the mitochondria of our cells, and we do a process called cellular respiration, which releases energy. We're going to be learning about that in the cells unit coming up soon. So, some examples of monosaccharides: glucose, fructose, galactose. One thing you'll notice, these are all 6-sided sugars. They all have the same formula, But the placement of atoms and the placement of things, the shape of this six-sided sugar will be different for glucose, fructose, and galactose. And therefore what they're called, so they have the same formula, but they are isomers of each other. An isomer means that they have a different shape, but they have the same formula. And what your body will do, so for example, if you're eating fructose, it's the same formula. Your body will use enzymes to actually turn it into glucose, that then, therefore, it can be used in cellular respiration. Because the shape of things, as we'll learn, is very important to these molecules, because our enzymes work based on shape. The shape of things, the shape of bonds and the shape of molecules is very important to our bodies. So that's some examples of monosaccharides: glucose, fructose, galactose, which is made by bacteria. Glucose is the product of photosynthesis. So plants make a lot of glucose, and then they use that glucose to make energy for themselves. And we eat the plants to make energy for us. We can't make our own energy, we gotta eat plant energy. So isomers, we said they have the same formula, but a different arrangement of atoms. That's the key here. So when you combine two monosaccharides, so you use dehydration synthesis, water is released, you've got a disaccharide now, and so some common disaccharides: sucrose, which I just talked about, which is table sugar, and lactose. Lactose is a combination of glucose and galactose, so bacteria and the plant one. And that is, lactose is the sugar that's in milk. Some of us don't have the enzyme to break that bond, so the shape is going to be different. Remember, galactose is an isomer of fructose, so it's going to have a different shape. So we have the enzyme to break fructose-glucose bond, but galactose, glucose, that galactose is going to be a different shape. So the enzyme that has to grab the two and break them and add water, remember, that's going to be hydrolysis, so it's adding water. The shape of galactose is different, so it can't grab the galactose. So there's a different enzyme called lactase that is for breaking those bonds, and we have it when we have kids, but we lose it over time. Some of us don't ever have that enzyme, and therefore we have trouble digesting milk. Essentially what happens, lactose intolerance, is that sugar, that lactose sugar, passes through our intestine and passes into our large intestine where water's sucked out, where a lot of bacteria live. And those bacteria then are like, hey, you're giving us sugar. And they digest that sugar because it's galactose, which is something that bacteria make, and glucose, and are able to metabolize it. And then they produce their wastes, basically, make you gassy and it can cause pain. Here we go. When 3 to 9, so 3 to 9, there it is, 3 to 9 monosaccharides. So we disaccharides, sucrose, lactose. When 3 to 9 of those glucose molecules, or whatever, glucose, galactose, or fructose are chained together, you get an oligosaccharide. And our body uses oligosaccharides. We make oligosaccharides. Oligosaccharides are actually on the cell membranes of our cells. And they're going to be unique. The combination of fructose, glucose, and how the bonds are made is going to be unique for each of us. And this is how our cells identify each other. We have many different oligosaccharides on our cells. So blood types, for example, A, B, C blood type, those antigens, those oligosaccharides are going to be different for A type, B type, AB type folks. Well, AB type folks will have both A and B type oligosaccharides on their cells. So that's where oligosaccharides are used for us. Those short chains, 9, what did I say? 3 to 9, those short chains are called oligosaccharides. And they're going to be used in cell identification. So know that. And then finally we have polysaccharides. So when more than 9 monosaccharides are joined together, you have a polysaccharide, a huge long chain of sugars, and we use this to store sugar. So we make glycogen. Our liver actually stitches glucose together when there's too much glucose in the blood, and your cells are just like, I'm just lying around. you're not doing anything. You don't need all that sugar for energy, and sugar is harmful in your blood. It's harmful in your cells. It can become harmful. So diabetics know this very well. So what happens is your liver is like, well, let's just take those glucose and stitch them together into glycogen and store it. And that's fat. You start to deposit fat. That's actually glycogen gets stored in fat cells and in your liver. When more than one, I repeat examples, and also glycogen. That's and so there's glycogen, there's starch, and there's cellulose. Starch is how plants store sugar, and cellulose, they don't actually, plants don't use, they use cellulose to store, but they use cellulose really to build structure. So they use sugar to build structure for themselves. So, like celery has a lot of cellulose in it, celery, and it has those long strings, that you chew on to get, you can't actually digest those, but hold on, I'm going to get to that. And basically what differentiates starch, which is the plant storage of sugar, and glycogen is how they're joined together, the shape of the bonds, basically how the molecules are arranged. So in glycogen, they branch sort of all the you can see, I don't know if you here, zoom in here. So starch is very branchy. I don't know if you can see that in there. Just look at the note, follow along with the notes, and you can see. So there's, every once in a while there's a branch. Glycogen, how that, starch is glycogen. So starch is spiraling. It's usually in a spiral. Starch is usually in a spiral, but so it's not as branched as glycogen. So glycogen is the way our body stores glycogen. Is it lots and lots of branches, so you can see here branchy, and then cellulose is basically straight lines. And so we can digest starch, obviously, because we love eating potatoes and things like that, and beets and carrots. Those are all starch, and pasta. Well, pasta's not anyways, doesn't matter. Potatoes are or rice is a huge example of starch, huge storage for starch. We can digest glycogen, but cellulose we can't, and that has to do with just the shape of the bonds, the shape of those molecules. Again, an enzyme has to come along and do hydrolysis to break those bonds. It has to add water, break that covalent bond that's holding those monomers, those glucose monomers together, and we don't have an enzyme that can do that for cellulose. Cows can. It's hard for them, but they can. They have multiple stomachs. They can do that. Some people think that the appendix, which is a vestigial part of our digestive system, which we'll probably talk about when we get to the digestive tract, used to store enzymes for digesting cellulose, but since we eat the animals that digest cellulose, we just don't need it. Sorry, animals, you will do the digesting of cellulose for us, and then you will convert that cellulose into glucose and other things, and we'll get that from you. Other sugars to talk about in the body. So these are just important sugars in the body. So just make note of those: sucrose, lactose. Those are disaccharides, glucose being the most important, which is a monosaccharide, and then fructose, which is an isomer of it, galactose, which is an isomer. No of those. No oligosaccharides are for cell identification, and then starch, glycogen, and cellulose. Starch and glycogen being the main storage, starch in plants, glycogen in animals for storage of glucose, and cellulose is used for structure in plants. And that we can't digest cellulose. Finally, there's two more sugars that we need to talk about. So without carbohydrates, obviously we couldn't exist. We need, because like I said, glucose ends up in our cells and it ends up being oxidized. It gets broken down and all of this kind of stuff happens. And that produces energy, produces something called ATP, and that process is called cellular respiration, which is very important. We're going to be encountering it. We're not going to be learning the details of cellular respiration, thankfully, but we're going to be encountering it throughout. I'm going to be constantly being like, why does this happen? And more often than not, the answer will be cellular respiration. So the process of cellular respiration is basically taking sugar in the presence of oxygen, breaking it down, producing carbon dioxide and water and ATP. And that ATP is our body's currency of energy. We use that for energy. We use it to do every other process. We use that ATP to make those covalent bonds. We use that energy to make those covalent bonds, to build structures, to build the larger structure of ourselves, proteins and all the other stuff. Now the other sugars, so those are 6 carbon, glucose, 6 carbon sugar. We just talked about 6 carbon sugar, 5 carbon sugars. There's ribose for our purposes. There's ribose and deoxyribose. Ribose is a multiple of the empirical formula. Remember I said the empirical formula, the most basic sugar carbohydrate formula is CH2O. A hydrate of water. So ribose is a sugar that is an empirical formula. It is C5H10O5. And you will find ribose in RNA. And RNA, we will learn about that when we do the genetics section, is very important. RNA is actually part of ATP, which is the currency of energy. It's actually a modified nucleotide of RNA. And then those nucleotides, which we'll talk about shortly, what RNA nucleotides are used for making proteins and storing genetic information. But the big storage of genetic information is DNA. And how DNA varies from RNA is just the sugar that's in the nucleotide, that's in the DNA molecule, is deoxyribose. And it does not follow the empirical formula. It has 5 carbons, but it's missing an oxygen. So it's C5H10O4. It's missing an oxygen, minus an O. Therefore it's deoxyribose, because it's deoxyed, doesn't have an oxy. And they are just part of the nucleotides that make up RNA and DNA. So there you go. Carbohydrates, very important because they help make up DNA and RNA. So there you go. That's sort of it for our purposes about carbohydrates. And in the next unit, I think, I can see there, we're going to talk about lipids.